my name is Ben Gilbert. I'm a student with Zana Luthi-Schulten at the University of Illinois, and today I'm going to be talking about the dynamics of chromosome organization in minimal bacterial cell. And so recently we published this paper, so if you're interested, you can learn more at the below paper. So the kind of topic at hand is the replication and inheritance of the genetic material in these minimal cells, because for any organism to proliferate, it must replicate its genetic material, and then it must ensure the inheritance of the replicated genetic material. So hopefully a kind of obvious question is, what is the simplest organism that can accomplish these tasks? One possible candidate for this is JCVI-SYN3A. So as you all know, it's synthetically minimized, so I'm not going to bore you with those details. So moving on, in our prior work modeling JCVI-SYN3A's chromosome, we started the cryo-electron tomograms, and from that we were able to reconstruct the ribosome distribution, and then we folded a lattice model of the chromosome amongst the ribosomes, and from that we were able to look at the overall organization of the genome by comparing it to 3C-seq done by the lab of Remus Dame at Leiden University. Unfortunately, that lattice model didn't really work for replication and segregation of the chromosome. So now we've instead moved to this continuum model where the entirety of SYN3A's 543 kilobase pair chromosome is modeled as a little over 54,000 beads on a string, where each of these beads contain 10 base pairs of DNA and are 3.4 nanometers across. And then the ribosomes are yet larger spheres that are 10 nanometers in radius. And so these beads then interact through a set of potentials that essentially represent and model the essential mechanical properties of double-stranded DNA. So first off, there's a bending interaction to give DNA its linear persistence length. There's a twisting and an aligning interaction to give it its twist persistence length. And then additionally, there's a stretching potential that controls how far the beads can stretch from one another. And so in addition to all of this, there are excluded volume interactions between both the DNA monomers themselves and then also between the DNA and the ribosomes and ribosomes and ribosomes. And those prevent the strands from crossing and ensure that the actual topology of the chromosome is maintained when there aren't topoisomerases present or other proteins that will modify the topology. And so just to quickly run through the challenges of carrying out this chromosome replication and segregation, first off, we have to know how we represent chromosomes in trivial replication states with nested theta structures, which I'll describe in greater detail shortly. Next, we have to know how the double-stranded DNA strands of the daughter chromosome are actually added to the system. And then finally, we need to know how the daughter chromosomes segregate while they're being replicated from the mother, because this is all occurring simultaneously. So to start with the non-trivial replication states, these are generally characterized by the ratio of the number of origin to the number of termini, where the origin is the origin of replication. And so this is greater than 1 in most cases for bacteria, and in SYN3A it's around 3.4. So to represent all these, we use this binary tree model, which I'll explain now. So here at the first point, you're viewing an unreplicated chromosome where the origin to termini ratio is 1, and it begins with the mother chromosome denoted M. It then begins replicating. The mother origin disappears because it's now been formed into the left daughter at a right daughter's origin denoted ML and MR. The origin-to ratio is now 2 to 1, and the total genome content, the total DNA content has increased by 30 monomers. And so this can continue, and we can carry out further replication where the origin-to ratio remains 2 to 1. And finally, we can get to these non-trivial replication states with these nested theta structures where the origin-to ratio is now 3 to 1, and the right daughter has now begun undergoing replication before it's finished replicating from its own mother. So the next challenge was how we actually replicate the daughter DNA in these kind of complicated structures, such as what you see on the left. And so we use this train track model of replication where you can imagine the DNA unzipping and the replication forks follow along the contour of the mother chromosome, creating the 2 daughter chromosomes.
Ben, it might be worth noting that the origin to terminus ratio was empirically determined as opposed to theoretically determined.
Yes, thank you, John. I failed to mention that. The origin to ratio was measured using qPCR to be 3.4 to 1. And so we believe, we're confident that that's correct. And so the third part is how we actually segregate the chromosome. So what happens between these states where it's kind of hopelessly tangled at the top, and then at the bottom we have it neatly partitioned where there are 2 daughter chromosomes and the cell is ready to divide. So the prevailing theory about how this occurs in all domains of life is that the segregation occurs via DNA looping by SMC complexes and strand crossings by type 2 topoisomerases. So there are also other proteins that assist in all of this and other systems that participate in all of this, but a lot of it is carried out by the action of just SMC complexes and type 2 topoisomerases, where the SMC create loops of DNA, and then the topoisomerase can detect where the DNA strands are crossed with one another and then pass the strands through one another. So interestingly, both SMC and topoisomerase are essential in Syn3A. So to investigate this further, we created a computational model of the looping by SMC complexes, where we have these two domains, this anchor domain and this hinge domain, that grab onto the DNA, and then essentially the hinge domain moves forward and extrudes loops of increasing size. And so based on that, we can show that the chromosome is uniformly compacted to a greater extent with an increasing number of loops, as we can see by the graph on the right. And then the computational model of the type 2 topoisomerases we accomplished by using a soft pair potential that's periodically switched on and allows the strands to cross each other when they're in tension. So we can see this example where we pull a DNA circle into a knot, so it's a trefoil knot, and with topoisomerase it becomes unknotted, but without topoisomerase it remains knotted no matter how much you pull on it. And so to look at the overall influence of these combined, we looked at 6 cases where we varied the number of loops, and then we varied the presence or absence of topoisomerase. So we did this for a toy system 1/10 the size of Syn3A. Throughout the simulation, we allowed replication to occur, and it was multi-fork replication. So initially it's unreplicated, then the mother begins replicating. So what you're seeing in this graph is the extent of replication, and then the left daughter begins replicating before the mother itself is finished replicating, before it reaches 1. And then the right daughter begins replicating at the end. So that takes us through 4 different replication states with varying Ori-to-Ter ratios, all the way from 1 to 1, all the way up to 4 to 2 in the last state, and then results in this final bond topology that you can see at the bottom, where we have 2 fully replicated chromosomes, both of which are themselves undergoing replication and making their own daughters. So to characterize how segregation occurs, we looked at the disentanglement of the system. So here are the 6 cases I described with varying loop numbers and with or without topoisomerase. And then we calculated the degree of disentanglement, which is what you can see me plotting. And that is displayed for every possible set of forks in the system. So qualitatively, it's immediately apparent that the bottom right case is the most segregated, where you can see kind of these distinct regions of different colors. But what we found that is interesting is that you need both topoisomerase and SMC looping to actually effectively segregate the chromosome in this case. If you just have looping and no topoisomerase, the first chromosome can get segregated to some extent, as you can see by the line labeled M increasing, but the 2 daughter chromosomes, ML and MR, when they start replicating, their 2 daughters actually end up even further entangled because they've already been compacted by the loops. And then you can see that if you just have topoisomerase without loops, the chromosomes, the DNA strands are never pulled into tension to actually resolve the strand crossings. So that's why you see with 0 loops, you don't see any disentanglement. And the same is true of the partitioning, where we're now looking at the distance between the centers of mass of the chromosomes relative to an ideal partitioning in the spherical cell. And we observe exactly the same trends, where you need both looping and topoisomerase to effectively segregate the chromosome. So now I'm going to talk briefly about chromosomes organization and how it's actually measured. So it's measured typically using 3C, where you cross-link all of the chromosome, and then you sequence fragments of it that are essentially connected then after they're ligated. And based on that, you're able to determine the frequency at which different regions of the genome interact. So interestingly, outside of some isolated cases in eukaryotes, these experimental methods can't actually distinguish between mother chromosomes and daughter chromosomes because they have the same sequence, of course. However, in my in silico map and these in silico maps that I've modeled that I've presented, if we calculate a chromosome contact map, it's actually possible for me to distinguish between the daughter interactions, the intra-daughter interactions of a daughter interacting with itself, and the inter-daughter interactions of a daughter interacting with another daughter of the system. So that's what you're seeing on the right. So the solid areas are the self interactions, and the checkerboard areas off the diagonal are a daughter interacting with another daughter. And these would be completely indistinguishable by experimental 3C methods. And so here's an example case where we can see how this contact map changes over time as the system undergoes replication. So we start with an unreplicated chromosome, and we only have the mother chromosome. And then as it splits into the left and right daughter, we can see that we start gaining these interactions off the diagonal from the daughters interacting with one another, which would be totally indistinguishable by 3C. This can continue as it continues to undergo replication, and you have a greater and greater complexity as there are greater and greater number of replication events. And so we can only compare this to experimental measurements if we then have a means to actually map it back to what is seen. So what we do is we take our true contact maps on the left, and then we convert them to sequence equivalent maps using this procedure that I'm showing where we sum up the different contributions. And these actually reproduce contact maps that show, kind of the standard behavior of a circular chromosome and the expected power law behavior. So finally, hopefully a lingering question for y'all is, does this actually work on the full Syn3A chromosome? Because I've only shown it so far for the toy system. And the answer is that it does. So here is the full Syn3A chromosome with 500 ribosomes. There are 100 loops acting throughout it. And we simulated the compaction by loops and the progressive replication and segregation of 20,000 monomers, which is about 40% of the chromosome. So here's the compaction, and then it slowly is replicated further and further. And so you can see the two forks in magenta advancing along the chromosome and the newly replicated chromosome in green. And then it continues going on. And as we advance, it becomes more and more disentangled, but the partitioning is still occurring slowly. So here we can see we've now fully disentangled the regions that were initially replicated in the blue and the yellow green, and then the replication has now passed along further into the chromosome. And so if we look at this quantitatively with the degree of disentanglement, we can see that what we observe qualitatively is indeed occurring. And so just to conclude, like I mentioned, if you want to learn more, you can go look at this paper. So in summary, we created this Brownian dynamics model. We are able to represent non-trivial replication states using our binary tree, and a train track model to actually generate the physical structures. And then we looked at chromosome segregation with computational models of SMC complexes and topoisomerase. So in terms of our findings, we found that solely SMC complexes and topoisomerases are sufficient to segregate chromosomes in Syn3A. We found that segregation occurs in parallel across all the generations of daughters when you have these nested theta structures, and that subpopulations of cells in different replication states alter contact maps from 3C methods, which motivates determining what the actual subpopulation should be, so that when you look at a 3C map, you can appreciate that it's not just a single unreplicated chromosome always. So in terms of future work, we want to couple this chromosome model to our whole cell model to simulate cell division. We're working on Martini simulations of an entire cell with the lab of Stuart Jan Marink, which I think Jan Stevens is going to talk about a little bit later. And then we need to actually determine what these multifork systems are and what their population should be by looking at kinetic modeling of multifork replication. So I'd like to thank all the following, and I'm happy to answer any questions.
Nice job, Ben. It's dazzling work. My first question is, what could we do empirically to test your model? My first thought is maybe could we increase the what would the effect be of increasing the concentration of SMC or the topoisomerases? So
I think obviously removing SMC or topoisomerase would be lethal, as is also confirmed by the essentiality. I don't know how SMC behaves once there's extra SMC there. So that would probably require in my model adding some kind of interaction between the SMC itself and modeling it more like a physical structure as opposed to using the harmonic bonds that kind of advance stochastically, which is what I'm currently doing. I think there are opportunities to look at to kind of extend what's been done with the qPCR and do that for a greater number of loci throughout the genome. And based on that, I think you could have further constraints to hopefully develop a mathematical technique to kind of deconvolve what the populations of the different replication states are. So I think that's one opportunity. And also looking at contact maps in greater detail, kind of knowing what we now have demonstrated is actually changing them based on the number of replicated daughters.
Thank you.